Okay. Good afternoon. My name is Marlene Arellano, and I worked in the plant genetics lab with Jeremie Fant. And I also worked on Castilleja cocinea, but I focused on its genetic diversity. So as you can see in this one, there are populations that come in like scarlet red color, but there are also populations that come in a yellow color. And it's a prairie plant that's found typically throughout like the eastern half of the United States. And it's a hemiparasite, which obviously means that it can photosynthesize, but it also feeds off its host plant, which are typically grasses. And with the Castilleja, an important part of it is what is known as pollination syndromes, which all that is the different characteristics or traits of a flower that affect what type of pollinator it has. So, for example, bird-pollinated flowers are typically a scarlet, orange, red, or white color, whereas bee-pollinated flowers are bright white, yellow, blue, and UV. So this means that obviously the Castilleja comes in a red and a yellow color, meaning that the red populations are pollinated by birds and the yellow by bees. And then we have the concept of genetic diversity. So all it is, it's a variation in the nucleotides, genes, chromosomes, or whole genomes of an organism, and they're called SNPs, which just stands for single nucleotide polymorphism. And there are many benefits to genetic diversity, specifically the fact that the flower has the capability to produce a wider range of genetically distinct offspring, which obviously helps the population as a whole be less susceptible to disease, more successfully survive, and pass on these traits to the next generation. So if we kind of bring these two concepts together, it made me wonder whether if in the species Castilleja coccinea, while having different pollinators and essentially the two different floral bracts colors, affect the level of genetic diversity when we compare the yellow populations to the red populations. And I had a null hypothesis, meaning that I thought that having offspring of two different colors and having offspring and where each color had its own pollinator basically would have no effect on genetic diversity, meaning that the yellow and the red populations would both be equal in their levels of genetic diversity. Basically, I had a very long list of materials. Some of the most important ones were my extraction kit, which basically I'll get to in the next slide. And then we had our leaf samples and a lot of different machines, but I used the centrifuge a lot. And basically the first step was to get your DNA from your leaf samples. So all you had was your extraction kit, which are a series of chemicals that basically isolate your DNA from like your RNA, your proteins, your polysaccharides, and all that other junk. And you end up with like your ready-to-use DNA. So then with that DNA, I would nanodrop it, and you kind of get back your nucleic acid concentration. And then I tested over like 200 leaf samples, and basically I picked the top 7 through 12 for each population, and I had 4 populations. I had the dropseed prairie, the sandridge, and those were my yellow populations. And then I had my Hoosier and my Shaw, which were the red populations. And then PCR was mentioned with the Enethera. And basically what I was using were the microsatellite genetic markers, and all it is are, repeating sequences of DNA. And it's used to kind of amplify it. So as you can see in this picture, you're taking like a sequence of DNA and you're amplifying it so that you have enough DNA to be visible and to use. And then obviously you have to compare your data between the red populations and the yellow populations. And basically this is kind of what my results look like, where I had the population, the color, your average variation at each marker, your genetic diversity, and your inbreeding coefficient. So for inbreeding coefficient, obviously it's a measure of how inbred your population is to one another. And the lower those levels, the better, because it suggests a higher gene flow. So just a quick glance at this, you can kind of see that they're all relatively low. And then I had this negative number, which all it means is that that population has more heterozygous than homozygous offspring. And then my main focus, of course, was genetic diversity. And the higher the number, the more genetically diverse. And keep in mind that these numbers are kind of relative to one another. And just a quick glance at this, you can kind of see that they're all around the same value. So if you put those on a graph, you can kind of see that there's really no big giant difference, but the red populations are a little more genetically diverse than the yellow populations. And then we had what was known as genetic distance, which is how genetically alike two populations are to one another. And some very interesting data was the fact that this yellow dropsy population was, in comparison to the other yellow population, was less genetically alike than that one was to the red population, which I guess all that can suggest is that it may not take much to change the color of a flower. And in the end, my hypothesis was not supported because though there wasn't a significant difference between the genetic diversity of the red and the yellow populations, the yellow populations were slightly less genetically diverse. And if you recall, the inbreeding coefficients were all relatively low, which kind of suggests that there are high levels of crossbreeding and a high level of gene flow between the populations. And it kind of gives us an indication that the genetic diversity of this species is still on the rise, which is a good thing because, as you recall, there are many benefits to genetic diversity, like the population's ability to, it's less susceptible to disease. And so in the end, we can kind of say that pollinators do not seem to have an effect on the genetic diversity of a flower at this point. Many thanks to all my mentors and Jocelyn and Robert, who kept me company in the lab and were a big help.